Inside your body, at this very moment, there exists an instruction manual so extraordinarily detailed that it contains nearly 3 billion characters of biological information. It is written not in words, but in a chemical language of just 4 letters. And for most of human history, we had no idea it was there. This is the story of how humanity finally learned to read the most powerful code ever created by nature, and what we discovered when we did. For thousands of years, humans bred dogs, crops, and cattle, noticing that children resembled their parents, that traits seemed to pass from generation to generation. But the mechanism remained utterly mysterious. Why did some traits skip generations? Why did children inherit combinations of features from both parents? The answer seemed to hide in a realm too small for any eye to see, in structures so minute that they wouldn't be observed for centuries. In the 1800s, an Augustinian friar named Gregor Mendel was cultivating pea plants in a monastery garden, and he noticed something extraordinary. When he crossed tall plants with short plants, the offspring weren't medium height, they were tall. When he bred these tall offspring together, some short plants reappeared in the next generation. Through meticulous observation of thousands of plants, Mendel began to see the outline of a pattern, the shadow of mathematical rules governing inheritance. He didn't know why these rules existed, he only knew that they did. His work went largely unnoticed during his lifetime, hidden in an obscure scientific journal. But decades later, scientists would discover that Mendel had stumbled upon the fundamental laws of heredity itself. The 20th century began its obsessive search for the physical basis of inheritance. Scientists knew that something in the nucleus of cells held the secrets of heredity, but what? In 1953, 2 brilliant minds, a young American named James Watson and an English physicist named Francis Crick, were trying to solve a puzzle unlike any before. They worked in a small laboratory at Cambridge University, but they weren't doing experiments. They were thinking. They were building models. They were staring at X-ray crystallography images made by a remarkable woman named Rosalind Franklin, whose contribution would later be overshadowed by history, though never by those who truly understood science. What they discovered that February morning would reshape the entire trajectory of human knowledge. The molecule of heredity, deoxyribonucleic acid, DNA, wasn't a shapeless blob. It was a double helix, 2 intertwined strands spiraling around one another like a twisted ladder. And in that elegant structure lay an answer that nature had encoded billions of years ago. The 2 strands were complementary. If one strand read ATCG, the other read tag C, they fit together perfectly. It was as if nature had written a sentence and then written the exact mirror image on a second page. This wasn't just beautiful, it was revolutionary. Because if you separated those 2 strands, each one could serve as a template to rebuild its perfect partner. This was how life copied itself. This was how life persisted across billions of years and billions of organisms, each generation reading the instructions written in the generation before. But understanding the structure was only the beginning. Scientists wanted to read the code itself, the human genome, the complete set of genetic instructions for building a human being. It contained somewhere between 3 and 4 billion base pairs. The letters A, T, G, and C repeated billions of times. And embedded within that sequence was everything needed to create a beating heart, a thinking brain, immune cells that could recognize invaders, eyes that could see light, ears that could hear music. Every human trait, every vulnerability to disease, every extraordinary ability, every limitation, it was all written there. For decades, reading even a tiny fraction of the human genome was a laborious, expensive process. Scientists would spend months sequencing just a few thousand bases. But then, in the 1990s, something remarkable happened. Technology accelerated. Computing power increased exponentially. New sequencing methods became faster and cheaper. And a man named Francis Collins, the director of the National Institutes of Health, had a bold vision. What if they didn't just read fragments of the human genome? What if they read all of it, every single base pair, the entire 3 billion letter instruction manual that made a human being possible? The Human Genome Project began in 1990, and it was unlike any scientific endeavor in history. Thousands of scientists from dozens of countries, working in hundreds of laboratories, united by a single audacious goal. They would map every gene, sequence every chromosome, and create a complete blueprint of human life. Rivals initially worked in competition. A private company led by Craig Venter and a public consortium of researchers led by Francis Collins. But by 2000, they jointly announced a landmark achievement. They had successfully sequenced the first draft of the human genome. The code of human life had been read. And then came the shock. Everyone expected that humans would have vastly more genes than simpler organisms. A worm had about 20,000 genes. But a fruit fly had about 14,000. Surely humans, with our complex brains and intricate bodies, would have hundreds of thousands of genes. But the data revealed something unexpected. Humans had only about 20 to 25,000 genes, barely more than a worm, fewer than some plants. How could this be? The same fundamental question that had driven science forward for centuries reared its head again. If our genes weren't responsible for all the complexity of human beings, what was? The answer came from an unexpected direction. Scientists realized that single genes didn't produce single proteins with single functions. Instead, the same gene could be spliced in different ways, creating multiple proteins with multiple functions. Genes could be turned on and off in response to signals from their environment. A gene wasn't a simple on-off switch. It was a dimmer that could be modulated infinitely, and above the code itself existed another layer of instruction. Chemical markers could attach to DNA, changing which genes were expressed and which lay dormant. This was epigenetics, the science of how genes could be turned up or down without changing the sequence itself. A single genetic code could create vast complexity through regulation, through context, through conversation between genes and environment. But the most profound implications came as scientists began comparing human genomes to one another. Every human genome is unique, yet they're also strikingly similar. We're more than 99% identical at the genetic level. That tiny difference, less than 1% of 3 billion base pairs, generates all the visible variation between humans: eye color, height, skin tone, hair texture, susceptibility to disease, intelligence, personality. It all emerges from that fractiona L gap in the code. Remarkably, humans share 98% of our DNA with chimpanzees, 95% with mice, 80% with dogs. The differences that seemed so vast, so fundamental, turned out to be written in the margins of a code we shared across all life. Darwin had been right about evolution, but he couldn't have imagined how precisely we'd eventually measure our kinship with every living thing. As geneticists began reading human genomes in detail, they encountered something deeply personal. They could identify the genes that influenced height, that affected whether someone had brown eyes or blue eyes, that carried the risk for certain diseases. This opened possibilities that seemed miraculous and terrifying in equal measure. Could we eliminate genetic diseases? Should we? If we could identify which genes contribute to intelligence, should we choose those genes for our children? What was the difference between curing disease and enhancement, between therapy and redesign? For the first time in human history, we possessed the ability to read the code of inheritance. And almost immediately, the next question emerged. Could we write it? The technology that would make this possible began developing in the depths of bacterial cells and microorganisms that had been battling viruses for billions of years. Bacteria evolved molecular scissors, systems that could cut DNA at precise locations and either destroy invading viruses or incorporate beneficial genes. Scientists observed these ancient molecular machines, enzyme systems called CRISPR, and realized something extraordinary. These tools could be reprogrammed. They could be aimed at any genetic sequence you desired. They could cut and fix DNA with unprecedented precision. In 2012, two scientists, Jennifer Doudna and Emmanuel Charpentier, published their vision of CRISPR as a gene editing tool for human cells. And what they described seemed almost too powerful to be true. A way to edit the human genome as easily as editing text in a document. Find the typo, the mutation causing disease. Delete it. Insert the correct version. Save. The simplicity was deceptive. The implications were staggering. Clinical trials began with extraordinary caution and hope. The first use of CRISPR gene therapy in humans was for sickle cell disease, an inherited blood disorder that had tortured patients for generations, causing excruciating pain, tissue damage, and early death. Scientists took cells from a patient's bone marrow, used CRISPR to correct the mutations causing sickle cell, and then reinfused those corrected cells back into the patient's body. The results were almost miraculous. For the first time, a genetic disease that had been essentially a life sentence could be treated, not with medications to manage symptoms, but with a cure that reached back to the root cause. The mutation itself was gone. But as CRISPR technology advanced, a more controversial possibility emerged. What if you didn't just edit genes in adult patients? What if you edited them in embryos, in the earliest cells of a developing human being? What if you could erase a genetic disease before a child was even born? A scientist named He Jiankuai in China attempted exactly this. He edited the CCR5 gene in human embryos, creating babies supposedly resistant to HIV infection. The scientific world erupted. It wasn't just that he'd edited human embryos without sufficient safety testing. It was that he'd crossed into territory that most researchers felt humanity wasn't ready to navigate. If you could eliminate disease genes, could you also select for other traits? Could you create designer babies, children, engineered for height, intelligence, athletic ability? The line between therapy and enhancement, between medicine and redesign, had become impossibly blurred. These weren't abstract ethical questions anymore. They were immediate, practical, urgent. What should humanity do with the power to rewrite itself? Some researchers began exploring a different approach to genetic disease, not editing the gene itself, but replacing its function. Gene therapy involved introducing a healthy copy of a gene into cells that carried a mutated version, essentially providing a backup copy that could do the job the defective one couldn't. A revolutionary therapy for spinal muscular atrophy, a devastating genetic disease that typically kills children before they turn 5, showed that a single injection of gene therapy could change the entire trajectory of a child's life. Children who would have faced progressive paralysis and death began to thrive. But these breakthroughs also revealed something profound about the nature of genetic disease. Many of the most devastating inherited conditions were caused by mutations in single genes: cystic fibrosis, hemophilia, Duchenne muscular dystrophy. These were essentially typos in the genetic code, single letters copied wrong, cascading into catastrophic consequences throughout problems. The body was far more interconnected, more holistic, more mysterious than anyone had anticipated. Meanwhile, other researchers were using the power of genetic technology for entirely different purposes. Ancient DNA, fragments of genetic material preserved in fossils, bones, and teeth, could now be extracted and sequenced. Scientists began reading the genetic code of extinct humans. A Neanderthal who had lived more than 60,000 years ago. Denisovans, another branch of human relatives, known only through their DNA. These weren't just fragments of ancient life. They were messages from deep time, telling us stories about who our ancestors were and how they'd lived. When geneticists compared ancient DNA with modern humans, they made a stunning discovery. Modern humans living in Europe and Asia weren't direct descendants of Neanderthals, but they carried Neanderthal DNA in their genomes, between 1% and 4%, depending on ancestry. At some point, tens of thousands of years ago, modern humans had interbred with Neanderthals. The genetic evidence showed that this interbreeding had given modern humans useful adaptations, genes that improved immune function, that helped them survive in new climates. In death, Neanderthals had given modern humanity gifts encoded in our DNA that persisted to this day. We carried our extinct cousins within us. This revelation reframed our understanding of human evolution entirely. We weren't a lineage that had simply outcompeted and eliminated rivals. We were a species that had absorbed genetic information from cousins and distant relatives, incorporating their innovations into our own genomes. Evolution wasn't just competition and natural selection. It was also exchange, integration, and the gradual emergence of new capabilities written into the language of DNA. As the Human Genome Project expanded and genetic technology proliferated, another field exploded with possibility: forensic science. For decades, detectives had relied on fingerprints and eyewitness testimony, but DNA was far more powerful. A single cell left at a crime scene, a drop of saliva, a strand of hair, contained a complete genetic blueprint. DNA profiling could match suspects to crime scenes with extraordinary precision. It could exonerate the innocent. The Innocence Project, using DNA evidence, freed hundreds of wrongly convicted prisoners. DNA didn't lie. It was the perfect witness, present at every scene, carrying an indelible record. But forensic DNA also raised troubling questions. Databases grew, storing genetic information on millions of people. How was this information protected? Who had access to it? If someone committed a crime, should their genetic information be added to a database automatically? What about relatives? Their DNA was similar enough that familial searches could implicate extended family members who hadn't committed any crime. The power to solve crimes came with the power to surveil, to invade privacy at the most fundamental biological level. Forensic science also revealed the hidden history of populations. Genetic ancestry tests became accessible to ordinary people. Millions of people submitted their DNA to commercial companies, eager to learn about their ethnic heritage, their ancestral origins. The data revealed migrations, mixing, and movements across human history that traditional historical records had obscured. It showed that genetic ancestry was far more complex and mixed than most people's understanding of their own heritage. A person who believed their ancestry was purely European might discover substantial genetic contributions from Africa, Asia, or the Middle East. Racial categories that seemed so fixed and real in human perception were revealed to be fluid and constructed. Biological reality didn't match the rigid social categories humans had invented. As genetic science advanced, it also began intersecting with artificial intelligence in unexpected ways. AI algorithms trained on massive genetic datasets could now predict disease risk with astonishing accuracy. An AI system could examine a genetic sequence and identify mutations associated with cancer risk, heart disease, Alzheimer's. Precision medicine moved from concept to reality. Instead of giving all cancer patients the same chemotherapy, doctors could sequence a tumor's DNA, identify the specific mutations driving it, and prescribe targeted therapies designed to attack those particular mutations while leaving healthy cells untouched. Cancer patients, who would have been given weeks to live, were experiencing complete remissions, yet the same technology raised new anxieties. If insurance companies could access genetic information predicting disease risk, would they deny coverage to people carrying genes for expensive conditions? If employers could sequence DNA, might they refuse to hire people genetically predisposed to early mortality or illness? Genetic information was simultaneously liberating and potentially oppressive, a key to better medicine and a potential tool for discrimination written at the most fundamental biological level.